I'm going to be talking today about functional traits, and so because I want to make sure we're all on the same page, I'm going to go through some definitions first, so I can make sure we all know what a plant functional trait is. So 3 decades ago, when physiological ecologists were the only ones really thinking about functional traits, we might have defined a functional trait as an attribute of a species that influences an individual's reproductive fitness. Okay, so these were traits that were directly linked to plants, in this case, plant performance. But in the last decade, lots of ecologists are now thinking about and using functional traits, and the definition has really broadened to something that really focuses on resource use and acquisition. So today, we might define a functional trait as an attribute of the species that relates both to strategies of resource capture and then also to the effects that that species has on the overall pool of the resources in the system. Okay, so this could be modified to encompass a bunch of different types of resources. And so really functional traits help us understand why particular species occur in the environment that they do, how they interact with each other, and then also how they can tolerate different environmental stressors. But probably one of the greatest advances that's been made in the last 10 years with respect to functional traits is how we've used them to understand plant growth strategies. So for example, we know that there's this axis of growth strategies for plants where on one end of the spectrum you have these rapidly growing annual species that have functional traits that all really pertain to rapid rates of resource acquisition and use. So they might have high specific leaf area. So this is just the amount of area per weight of a leaf, right? So the thinner the leaf, the more sunlight that's trying to maximize so that it can assimilate more carbon. And then on the other end of the spectrum, you have more slower growing perennial woody species that have the opposite suite of traits. Okay, so they tend to have really thicker tissues that live a long time so they can really maximize their investment in that tissue. And they're often really, rather than focusing on resource acquisition, they're all about resource conservation. So they might have high water use efficiency or high nutrient use efficiency. And then there's a bunch of species that are somewhere in the middle of the spectrum. And so this has really allowed, in particular, community ecologists to go out to the field and measure a couple of traits and be able to understand sort of the diversity of strategies that you might have in a system and then be able to make predictions about how that system might respond to changes in abiotic and biotic factors. So functional traits can just generally be used to describe or explain a bunch of processes across scales, from the organism to the community to the ecosystem. My work actually does cross a lot of these scales, but the work that I'll be focusing on today on invasion and restoration ecology is really focused at the level of the community, using functional traits to understand competitive outcomes, and then also the process of community assembly. All right, I'm also going to be talking about invasion. So sorry for those of you who came to my talk yesterday, this slide might be a review. But I wanted to just define an invasive species as one that spreads out of an area of introduction, reaching high local abundance at each site. Okay, so it's a definition that might not be very straightforward to some of us, but these are non-native plants and animals that we've introduced and they spread on their own once they get into that introduced habitat. So this definition doesn't necessarily have anything to do with that species having a detrimental impact on the system or species. It's just a plant or animal species that's not native to an area and spreads. But invasive species can have really deleterious impacts on systems, like they can decrease native plant biodiversity, they can change ecosystem functions like rates of carbon cycling, or they could destroy habitats or wildlife, that kind of thing. So lots of ecological restoration projects are now focusing on trying to get rid of invasive species and figure out ways to promote the growth of native species. So what is ecological restoration? That is simply the process of assisting the recovery of an ecosystem that has been damaged, degraded, or destroyed. And there are a lot of different types of ecological restoration projects. So there are some that are really simple. The 2 examples I like to use are ones where you go into a system, you make one modification, and then you walk away. There's no follow-up management. So for example, you might go in and remove a dam to restore normal flooding conditions, or if you know that your invasive species seeds are being brought into a system by being stuck to ungulates, you might put up fences to remove ungulates from that area. And then on the other end of the spectrum, you have more complex restoration projects. These ones might be where you're having to restore soils that have been contaminated, or you're having to go in with bulldozers and remove weeds, plant natives, run irrigation lines, and then do a bunch of follow-up management. So typically these more complex ones are more labor intensive, they often require more money and longer time periods. And the approaches that I'm going to be talking about today are really more appropriate for these complex restoration projects where you're essentially having to build a native community from scratch. So how might you implement functional traits into restoration protocols? I'm going to walk you through this slide because there's a lot of stuff going on. But this was a framework that some colleagues and I developed in 2008, and I've essentially been testing it ever since. There are still very few empirical tests of this framework. So a lot of the stuff that I've been learning about in the last 24 hours actually is testing this framework, which is very exciting. All right, so the native plants are in blue, the invasive plants are in pink, and each one of these different symbols corresponds with a different type of species. So the stars might be grasses, the circles might be leguminous shrubs or something like that. And you start out at a large spatial scale with a regional species pool, and you go through a series of filters until you end up in a local community at a much smaller scale. And over here on the left, we have our unassisted, degraded site, where most of the species of the local community are invasive. And over here on the right, we have our restored community, where we've actually gone in and managed it, and at the local community, we have all native plant species. All right, so in order for species in the regional species pool to get to the local community, they have to pass through these three filters. The first is a dispersal filter. So if a plant species can't get to the local community, it can't exist there. Okay, so first it has to be able to disperse to that site. Once it gets to a site, it has to go through a resource filter. So you could also think of this as an environmental filter or an abiotic filter. So all the environmental conditions have to be right for the species to exist there. So for plants, this is water, light, nutrient availability. You can also put in temperature, soil pH, other types of abiotic factors in this filter. And then if they can't exist, then they have to go through a biotic filter. This is primarily competition from the resident plants. It can also be things like enemies, so herbivores, pathogens, that kind of thing. So they have to be able to withstand those biotic interactions. All right, so how do we use functional traits? Based on our understanding of differences or similarities in functional traits between the native and invasive plant species, this might suggest ways that we can modify these filters to let the native species through, but to block the invasive species from moving into that local community. Okay? So one way you might modify the dispersal filter is an example I presented in the previous slide, where you know that the dispersal pathways for your native and invasive species are different. So let's say the natives are wind dispersed, but the invasive species are hitching a ride on ungulates into your system. You put up a fence, you exclude the ungulates. That's now, we've modified that dispersal filter into that community. Resource filters are a really great way to solve this problem, because oftentimes invasive species have higher resource requirements than native species, which are adapted to sort of pre-disturbance levels of resources. So humans have done a really great job of increasing resource availability in a lot of systems, like the wetland systems I've been learning about over the last 24 hours, which receive a lot of nutrients through agricultural runoff. Biotic filters, this is like probably one of the best ways that you can think about this is trying to restore a community by putting in really competitive native species. So once you restore the system, if you have really great competitors there, if these invasive species try to get back into the system, it'll be harder for them because they'll be experiencing greater competition from the native community. You could also put things like biological control, like if you were actually introducing some sort of enemy of the invasive species into that system, that might be a way to manipulate that filter as well. Okay, so my talk today is really going to focus, I'm going to talk about 2 case studies, some work that we've done in my lab, where we've first modified a resource filter and then modified a biotic filter based on our understanding of plant functional traits. Again, with the goal of creating native-dominated plant communities. All right, so I'm going to talk about the first experiment now. And this is based on this idea of the fluctuating resource hypothesis, which was posited a long time ago now, in 2000. And that's that invasion is facilitated by high resource availability, resulting from either disturbance or from low resource uptake by the native plant community. Okay, so the idea behind this theory is that we've added a lot of resources to systems, and that's facilitated the invasion of these high resource, these like nitrophilic species, for example. So if we can lower the resource availability back to certain pre-disturbance levels, then that should suppress the growth of natives, but not have any effect on the growth of native plants that are really used to these lower resource conditions. So some colleagues of mine, Robert Steers and Edie Allen, who at the time were at UC Riverside. Robert has since moved on, and Edie is actually retiring this year. We decided to test this in an annual desert community. We're working in the Morongo Preserve, which is just outside of Joshua Tree National Park, if any of you have been there. And what's happened in Joshua Tree and all the surrounding desert is that they've received a lot of nitrogen pollution from nearby Los Angeles basin. So a lot of car exhaust and industrial pollution gets put up into the atmosphere, and then the way that the winds blow, they blow all of that into these pristine deserts, which it's then deposited via dry or wet deposition into these remote desert areas, increasing the nitrogen availability of the soil. So our hypothesis was if we could figure out a way to reduce the nitrogen availability in the soil, that this would suppress the growth of invasives in the system. So this is a picture of our site right before a huge fire occurred, right before our experiment in 2005. And you can see that the canopy, if you want to call it that, is creosote bush and Joshua tree. And we have this very dense understory of this invasive grass, Bromus madridensis. In 2006, right before our experiment started, a fire went through the area. And a lot of the creosote and Joshua trees were able to recover, but the fire did shift the understory composition dramatically, and we now had this understory of Schismus barbatus, which is another annual exotic grass. So we still had the same setup, but the players just changed. So we decided to manipulate the soil nitrogen availability by adding carbon to the soil. And the way that this works is that when you provide soil microbes with this carbon source, they start breaking it down. But they need to maintain their own stoichiometric relationships of carbon to nitrogen in their cells. So for all the carbon that they're taking from the carbon source, they then have to pull nitrogen from the soil. And when they do that, that process is immobilization, and they lock that nitrogen up in the microbial biomass pool, making it unavailable for plants. And this is a very temporary process because those microbes will die, and when they decompose, the nitrogen is returned to the soil. But it's a really great way of locking the nitrogen up in the microbial biomass pool for a short period of time. So we created 3 treatments. We had a control treatment. These were just plots that we laid out here in the desert. One was just a control. There was no carbon that was added. We had a low carbon addition experiment or treatment where we were shooting for a 50-to-1 ratio of carbon to nitrogen. And then we had a high carbon addition treatment where we're shooting for a 100-to-1 ratio of carbon to nitrogen. And we always added the carbon in the form of sugar because that's very easy for microbes to break down, and it's very easy for us to access. And we always added it during the first rain event once a year, and we did this for 3 years. And the reason why we did it with the first rain event is that we knew that the invasive grasses in this system germinated really quickly with just a very small amount of rain. And so we wanted to hit them with this big nitrogen depletion event right when they were starting to establish. In contrast, the natives, they typically, they're more conservative. They want to wait until a bigger rainfall event or make sure that the soil stays a little bit wet for 2 to 4 weeks, and then they usually germinate a little bit later. Okay, so we assume that the natives might be totally unaffected by this nitrogen depletion event. But even so, even if they were exposed to this lower nitrogen concentration, they should be used to that, right? Because they were occurring in these systems before they were disturbed. So these are the 14 species that we collected a bunch of functional trait data on. So we had 3 invasive species and 11 natives. It's actually really diverse. There's a lot of native biodiversity in the system, but I put the percent cover data up here so you can see that the natives just really aren't that abundant. And in fact, the dominant invasive grass, the Schismus barbatus, was by far the most common species in the system, followed by Erodium secutarium, which is an annual forb, and then Bromus madridensis, which used to be the dominant grass in the system before the fire. The species that have these asterisks next to them, we actually have detailed biomass data for those 3, and I'm going to show you those data later. And we selected the Connactus just because it was the most dominant native of all of the ones that we measured. So we focused on functional traits that pertain to nitrogen and water use and acquisition. So most of the time, growth in these systems is going to be limited by water. But when water is available, then nitrogen is probably going to be the resource that is most limiting to plant growth. So we looked at root to shoot ratio. This is just the biomass of root compared to the shoot, and it really tells us a little bit about how the plants are prioritizing acquisition of resources. So plants in the desert typically have larger root to shoot ratios than they would in a wet area because they're mostly limited by below ground resources. Leaf thickness, leaf mass per unit area, and photosynthetic rate are all related, and they all tell us where the species falls along that axis that I talked about earlier, where you have some plants that have really kind of thinner leaves that are focused on photosynthesis, and then you have some species that have these longer lived tissues that are more about conservation and less about photosynthesis. We looked at water use efficiency, which is just the amount of carbon a plant can take up through photosynthesis per amount that it's losing, per amount of water that it's losing through the stomates. We looked at leaf nitrogen content, and we used this as a proxy for how much nitrogen a plant actually needs. Typically, these very nitrophilic invasive species tend to have high nitrogen contents, and that reflects their need for that additional nitrogen. And then we looked at photosynthetic nitrogen use efficiency, which is just the amount of carbon being taken up per amount of nitrogen in the leaf. And plants can change this, can alter this by preferentially distributing nitrogen to photosynthetic enzymes. So they have choices of where they allocate nitrogen in the leaf, and sometimes those choices can increase photosynthetic rate. All right, so here are the biomass data. I'm presenting data for 2006 and 2008, but not 2007, because that year we only had 1.2 centimeters of rain over the entire growing season. So nothing germinated. It was a really unfortunate time to conduct this experiment. But the 2 years of data were enough for us to see some really cool patterns. So this is percent cover, and these are our 3 treatments. So this is our control, our low carbon addition, and our high carbon addition treatment. This was Schismus. This is that dominant exotic grass. This is the Erodium. This is an exotic forb, and this is Connactus, the native forb. So in 2006, everything went as according to plan. We got a couple of small early rain events which caused the Schismus and Erodium and all the invasives to germinate first. They were hit with this nitrogen depletion event. I'm not showing you any of the soil nitrogen data, but we did manage to successfully decrease the soil nitrogen content. And that reduction led to this big decline in the percent cover of Schismus. For whatever reason, maybe it was like temperature cues or something, but Erodium just wasn't all that dominant that year. And the native plants largely were unaffected. Okay, so we ended this year thinking this is a really successful strategy. You add sugar to the soil, and it cuts down on the cover of Schismus. And we repeated this experiment in 2008, and we found a really different pattern. So what happened in 2008 was that we had a couple of really big rain events in the very beginning of the season. And so we didn't see this temporal distinction in germination times between the native and invasive species. Basically, everyone germinated at once. Okay, so all of these plants were then hit by this nitrogen depletion event. Okay, so we saw a decrease in the cover of all of the species. Okay, so this actually was puzzling to us because going into this experiment, we thought the native plants, even if they did experience a reduction in soil nitrogen availability, they should be okay. But when we actually went and looked at the functional trait data, There were no statistically significant differences in any of the nitrogen traits between the native and invasive species. So I'm just showing you the leaf nitrogen content data for these 3 species. But definitely the invasive species in that system did not have these really high nitrogen concentrations that we would normally expect to see in other invasive species. And in fact, some of the natives, in particular the ones that had really high water use efficiency, had very high leaf nitrogen contents. And so people have seen this. There's been a lot of work in Australia and other areas, semi-arid areas, where this is a really effective strategy for a lot of species that would like to increase their water use efficiency. So one way they do that is they do pump a lot of nitrogen into their leaves, primarily into those photosynthetic enzymes. So when water is available, they can open their stomates and photosynthesize like crazy. So if we had actually looked at the trait data before we started this experiment, we might have expected that if everybody was germinating at the same time and everybody was exposed to this nitrogen depletion event, that you would see this sort of effect. So we also looked at a bunch of the water use traits, which we found some interesting patterns there. So this is a principal components analysis. Essentially, it just takes a bunch of multivariate data and collapses it into 2 axes. It's really helpful when you're working with a bunch of trait data because it shows you which traits explain most of the variation across the species. And then it kind of allows you to see how the different species fall out in this trait space. So you can see which species kind of cluster together with similar traits. So we found that carbon assimilation traits explained about 39% of the variation among our species. So this just means that species on this side of the axis tend to have higher values of those traits. The second axis explained another 19%. Species up here had high root-to-shoot ratios. This species down here probably drove this water use efficiency response. But what we found was that the invasive species, they clustered together. They had low photosynthetic rates. They didn't have very good allocation to below ground biomass, and they weren't particularly water use efficient. So to me, this suggests that maybe these invasive species are making a bunch of cheaply constructed leaves, but they're making a lot of them. So they're diluting their leaf nitrogen content, they're making a lot of leaves so that they can photosynthesize, have higher photosynthetic rates at the plant scale. And this is a really good strategy in a wet year. But in a dry year, this I think would be a really bad strategy. So this data led us to get really excited, because Southern California is supposed to get drier over the next 50 to 100 years. And so we're like, maybe this is good news for the natives, right? The natives are really great at increasing their water use efficiency. They have high root-to-shoot ratios. If it gets drier, they'll be fine. But these invasive species are not going to be fine. Here's a data slide that I showed yesterday in my talk. Which basically shows that short-lived invasive species are able to adapt to climate change. So just to very quickly go over this slide, we had gone and collected some seed from 3 different water treatments. So an ambient, a reduced water treatment, and a water addition treatment. These were from a totally different invasive species, Avena barbata. So these plants were exposed to these different water conditions for 5 years. We grabbed the seeds, took them back to Chapman University and grew them for a couple of years just to get rid of any maternal effects. And we actually found that there were genetically linked phenotypic differences after just 5 years of growing in these different water conditions. In fact, the plants in the ambient and the reduced water treatments were able to flower earlier in the year, which is really advantageous as it gets drier because that ensures that you complete your growing cycle and including seed production before it gets too dry that you potentially die. So these data suggest that some of these short-lived, annual invasive species will be able to adapt to climate change, which was very disheartening to us based on the results that we found in this other experiment. All right, so just to conclude from this nitrogen manipulation experiment, we found that the nitrogen manipulation was an effective restoration strategy in the system, but only in those normal years when the invasive species germinates first, because the natives do get affected by that nitrogen depletion event. We did see that native and invasive species differed with respect to some of these water and carbon use traits, which got us, again, really hopeful that they might be be deleteriously, or they might be negatively impacted by climate change, but that we need more data on that. And then whenever I do a restoration project like this, I always like to step back and say, are these manipulations practical? Can we fill a crop duster full of high fructose corn syrup and fly it over the desert? Would that work? I've actually done some back of the envelope calculations on this, and it would be really expensive. And you'd have to do it every year, right? Because you lock up all of the nitrogen in the microbial biomass pool, but then it just gets returned back to the system. So this isn't feasible on a large scale, but it definitely would be feasible on smaller scales. So let's say you are working in Joshua Tree National Park, and you're working in like a sensitive ecological area, and you can't use herbicides, you can't introduce a biocontrol agent. Maybe these are like inaccessible areas where it's really hard to get like a field crew in to go pull weeds. This might be a really great approach. And it is fairly cost effective. I mean, you're basically just spraying a bunch of sugar all over the desert. So I think this could be a pretty practical restoration tool, but just on smaller scales. All right, so I will move on to the next case study. And in this one, we altered a biotic filter with the goal of restoring native plant communities. All right, so the theory behind this project is that of limiting similarity. So this posits that there's a finite limit to the similarity between the resource use of coexisting species. Okay, so I have a woody perennial here and an annual grass here. Okay, so these species might be able to co-occur like they do in a lot of the coastal sage scrub species or systems that I work in Southern California, because they're using resources in different spatial areas and then also at different times. Okay, so the grasses have these really shallow fibrous root systems. They're primarily getting water from these upper layers of the soil. These perennial species have roots that can go down 2 meters, and some of the data that I have suggests that a lot of their absorptive roots are actually further down in the soil. There's just a lot more water down here. It's more consistent throughout the year. So these species might be able to coexist because if they are competing more strongly for water, they're grabbing it from different areas. They're also active at different times of the year. So these annual species in California at least are active between November and May, whereas both the drought deciduous and the evergreen species can be active for much longer, like even year round. So it's quite possible that if we do, let's say this is an invasive grass species, it might be doing really well in the system because it doesn't have any species that are competing directly with it. Okay, so the idea of using limiting similarity in restoration is one where you try to go into the regional species pool and find other native species that are very functionally similar. So they're using resources in the same way as the invasive species so that they will provide more competition for those invasive species. So our research question was, can we build invasion-resistant communities by planting native species that are functionally similar to the invaders? And in particular, we were looking at these resource use traits. And this project was done in collaboration with Amy Wolf, who's now a postdoc at Columbia University. So we were working in the Serpentine grassland system. We were working at Jasper Ridge Biological Preserve, which is the field station for Stanford University. Probably you have serpentine soils in Utah. Is that true? Anyone know? Serpentine soils are incredibly cool. They have really high levels of toxic heavy metals. They have really high calcium to magnesium ratios and very low macronutrient concentrations, so low concentrations of both nitrogen and phosphorus. So they're really, really hard places for plants to grow. And because of that, we have all kinds of endemic species that you find nowhere else in the world, and all kinds of really cool adaptations that plants have to deal with these really harsh soils. Because of that, they've remained fairly uninvaded until recently, and we now are starting to see movement of a lot of these exotic annual grasses into the system. It's quite possible that these species have evolved to be able to tolerate with these soils, or maybe we've just had a new influx of different genotypes that are more equipped to deal with this. It's unclear. There are people working on that question, though. So at Jasper Ridge, one of the dominant invasive grasses is Festuca perennis, also called ryegrass, and that was the focal invasive species that we were using in this study. Our understanding of this system before we started this experiment was that in dry years, plants are competing primarily for water, but in wet years, they're competing primarily for nitrogen. So our specific research question was, is ryegrass growth reduced by native species with similar water and nitrogen use traits? So the project had two parts. The first was to go out into the regional species pool and actually measure the functional traits to identify which species were similar and which were dissimilar. And then do an actual experiment where a competition experiment where we grew functionally similar and dissimilar native species with the ryegrass and then looked at the competitive performance. So these are all the species in the regional species pool. I think it was something like 53 species. Again, a really, really diverse community. We have annual herbs, grasses, legumes, perennial herbs, bulbs, and grasses. All of the species that have these astraces are actually also invasive species, but we wanted to include them because it's quite likely that another invasive species is the one that's the most functionally similar. And if that was the case, we wanted to include that in our experiment to see if it did have the biggest effect on the ryegrass. So we found individuals of all of these species and measured a bunch of functional traits. And this list is very similar as it was in the first project because we were really focusing on traits pertaining to water and nitrogen use. So again, we looked at root to shoot ratio. This time we also looked at rooting depth, right? This is telling us where the plants are drawing the below ground resources from. We looked at specific root length, which is just a measure of the thinness of the root. So the thinner the root, the more surface area to volume ratio it has. And so for a given amount of material invested, it's actually more efficient at acquiring those below ground resources. And then we looked at leaf nitrogen content, again, as a proxy for how much nitrogen the plants need. We looked at leaf mass per unit area, as well as photosynthetic rate, again, to tell us where along that carbon assimilation axis these plants landed. We also looked at water use efficiency and nitrogen use efficiency. Here's another principal components analysis. So 35% of the variation in the species was influenced by leaf nitrogen content, and A stands for photosynthetic rate there. Another 22.5% was influenced by leaf mass per unit area and root depth. And what we wanted to do here was to use this analysis to see which species were the most similar and dissimilar to our focal invaders, which was the ryegrass here. So what we did was we calculated Euclidean distances, okay, which is basically just the distance in this trait space between all of the potential competitors and this ryegrass. And so we chose some that were similar and then some that were out here, so some that were dissimilar. So ultimately, we ended up with 5 functionally similar and 5 functionally dissimilar species. As we predicted, another invasive species, Bromus radiaceus, was the most similar. So these are actually the Euclidean distances. So as it gets closer to 0, that's the most similar. And actually, the most dissimilar one was a perennial bunch grass, Elmus multicetus. So for functionally similar, we chose this one exotic. I mean, we would never use this in a restoration project. This was just out of curiosity, is this going to be the best competitor? We had 2 perennial bunch grasses and then 2 annual herbs. So they are pictured here. And for functionally dissimilar, there were a lot of different species that we could have selected because there were a lot of species that were dissimilar. But we ended up choosing these ones because they're often used in restoration projects. They are very common in undisturbed serpentine grassland, and sometimes they perform really critical services. So for example, Plantego erecta, which is pictured here, is the dominant food source for the endangered checker spot butterfly. And so we were really curious to have some competition data for these species that we could give to restoration practitioners and say, maybe you should stop planting this species and instead plant this one because it competes better with the invasive species. So we grew all of these competitors in these tubes that had really narrow diameters, but they were very deep. We didn't want to prevent root growth into the soil. And they were filled with serpentine soil, and they were planted at a density of 20 plants per pot, which scales up to about 2,500 plants per square meter, which might seem really dense, but that is the actual density that these plants grow in the field. They're really, really tiny. And so that ended up being, 20 plants per pot. So they were either grown in monoculture or in mixture with the ryegrass. We grew them for a growing season, and then we harvested just the above ground biomass and weighed that to determine competitive outcomes. It would have been really hard to harvest these and get any root biomass, in case that's somebody's question. That would have been really hard. All right, so here are the performance data. So on the y-axis here, we have the percent of ryegrass biomass. So if it's negative, that means that the competitor suppressed the growth of ryegrass compared to when the ryegrass was just growing by itself, okay? And the competitors are arranged in the order of decreasing similarity. So we had the most similar bromus on the left and the most dissimilar native species Elmus on the right. Okay, and so if limiting similarity is to be believed, we would have expected to see a relationship something like that, okay, where the more similar species are leading to greater reductions in the growth of the ryegrass. But we did not see that, okay, so we did not see that at all. What was really interesting, and I didn't point this out earlier, is that we included a congeneric native, Festuca microstachys, which is actually this pretty short-statured plant. It's kind of this wimpy plant. We didn't think it was going to do anything. But it somehow managed to decrease the growth of the invasive ryegrass quite a bit, which is really interesting. So this led us to kind of go back into the data and try to figure out what traits did correlate with competitive ability. And what we found was that species that had high root-to-shoot ratios were actually really weak competitors. Okay, so another way of saying this is that plant species that put a lot of biomass above ground were really good competitors. So maybe they were shading out the ryegrass. That's one potential mechanism. Species that had a lot of leaf nitrogen in their leaves were really poor competitors. Okay, so kind of going back to some of the results from that first project, it's quite possible that species that were diluting their leaf nitrogen content and just making more biomass above ground, again, perhaps to shade out ryegrass, maybe they were the better competitors through that mechanism. We also found that specific root length was positively correlated with competitive ability. So again, that's just thinner roots, you're more efficient at acquiring those below ground resources. So this is totally in line with what we were expecting. So this led us to believe that maybe in this system, just being similar to the invasive species doesn't really matter. Instead, it helps just to have these trait values that are really important in that system. And it really got us thinking that maybe this whole assumption that we had that species were competing for below ground resources was false. Maybe species are also competing for light. I mean, they are growing, even though they're very tiny and short-statured, they are growing in really high densities. And so maybe shading each other out is actually very important. All right, so to conclude from this part of the talk, our data suggested that maybe there is no support for limiting similarity. I do want to throw out the caveat, I'm just going to skip around here, that we only measured traits really that pertain to water and nitrogen use, right? We weren't thinking about other traits going into this system based on the literature. We didn't think that light was going to be all that important. The fact that we found that a congeneric native was a really good competitor made us think that maybe there's some really important biotic interactions that we didn't measure. So for example, maybe the two closely related Festucas share some sort of pathogen, right? And so that's actually what contributed to the suppression and the growth of ryegrass. So there's just a bunch of things we didn't measure. We also, for reasons I won't go into, had to plant all of these species out as seedlings all at the same time. And so we didn't actually allow for any differences in germination timing to occur. So it's quite possible that phenological traits might be important. So this is actually one of the big problems of using functional traits to do anything in ecology is that, it all depends, it's very subjective. It's all about what traits that you're using. And so traits that might be important in one system might not be important in another. And it's really hard to determine which traits are going to be important. So competition for light might be more important than we really thought. But instead, our data suggest, rather than limiting similarity, that this idea of a competitive trait hierarchy might be important. There's been a couple of papers published on this in the last few years. And all this means is that for a given system, it really doesn't matter if a species is similar or not. They just need to have these values of traits that make them more competitive in that particular system. So in our system, it seems that plants that have low root-to-shoot ratio, high specific root length, but low leaf nitrogen content tend to be the really good competitors with Festuca. Probably those 3 traits would not be applicable to another ecosystem. My guess is that it's going to be very case or context dependent. All right, so I just wanted to come back to this basic framework just for a second. And I hope that I've convinced you that functional traits are interesting, at least, if not totally useful. Again, I welcome more empirical tests of this framework, and I know that some of you in the room are doing that, which is exciting. Again, I think that it's a good framework to use, and perhaps, we'll get to this point where some of these, filter manipulations might be an important way of thinking about how to restore communities, but the functional traits that you use might really differ depending on the system. There are a lot of people I need to acknowledge who helped me collect all of these data. So these are all the folks that I collaborated with who gave me access to sites or collaborated on the ideas. There's also a lot of undergraduates at Chapman who helped me collect the data and in particular process samples, which is very important. And I had a lot of funding sources for the 2 projects that I presented to you. And thank you very much for coming, and I'll take questions if you have them. Yes.
So at the end of the first section, you had an ordination, and then you had the 2006 and 2008 plots. And one of your, I think it was, maybe it was 2007, 2009, one of your take-homes was that, I'm trying to remember what you said, something about if it was drier, you would expect to see a different trend?
This one?
Yeah.
If it was drier? So the main difference between these 2 graphs is that this year we had like the typical precipitation pattern where we had just a few very small rain events early in the season. And so that's when we had the germination of the exotic species, but then the natives germinated like 2 to 4 weeks later. And then this year in 2008, we had these really massive rain events, and then everybody germinated at the same time.
So I know you left it out for a reason, but what did you learn from 2007 when it was really dry?
Yeah, nothing germinated.
Just nothing germinated?
Yeah, nothing germinated. It was the weirdest thing. I mean, it really was like a desert. There was nothing out there.
Even the invasives?
Yeah, I mean, probably like where you had these little like crevices and like, some of the rain was able to pool, right? There was some, but for the most part, there wasn't enough for us to even use for the experiment, yeah. Yes?
So I know it's hard because a lot of this work is like so context dependent and exactly the site. But with this work with ED that you did out in the desert, I know like it's really hot, so you have these like huge nitrogen pulses where you're using all of this nitrogen in the soil that you've allowed the microbes to access with more carbon. So would that work like on like a coast to desert gradient where it's a little bit more mesic, a little bit like wetter year round kind of and not as hot? Or like is this I guess like is this approach applicable anywhere besides like a really hot desert?
Yeah, I think it would be.
Okay.
So there have been other people that have used this technique. I'm trying to think of where Dana Blumenthal tried this. I think he works in Colorado. Does somebody know where? Yeah, I'm not exactly. Yeah, so I think that he's done it in a more mesic short grass steppe. I'm not familiar with that system, but yeah, I think that it has been employed in other systems. I don't know if it would work in a wetland, for example, but yeah, I think it could be used. I don't know if like the temporal dynamics would be the same. So we basically saw this big nitrogen depletion event, and it was basically done after like 2.5 months. So our soil nitrogen data came right back up to where they were. So I don't know if like the amount of water in the system and the temperature in the system, I mean, clearly that's going to control microbial activity. So I don't know if that would actually lengthen it or shorten that window. But those are good questions. Yeah?
I'm wondering about with your second experiment about the choice of traits,
Mm-hmm.
and this is a little bit more of a broader methodological question, but you said after the fact that there were other traits you could have measured or should have measured. For students that are trying to link trait data to other things, what's the right approach? Do you just try to measure the whole world in terms of functional traits, or still try to have those hypotheses ahead of time, and sometimes you might hit it and sometimes you just
Yeah. Well, so one way to do it, which I think I'll put Rachel on the spot. One way to do it is like when you're actually thinking about your restoration outcomes, right? So like if you are actually trying to create wildlife habitat, right? And one of the things that you're really interested in is like stem density. Is that right? Is that one of the things you're going to look at? Stem density, right? Like that might be a functional trait that you use, because that like is directly related to like the restoration outcome that you want. In this case, we were going for competition. And so that's why we focused on these nitrogen and water traits. I think a lot of community ecologists who are doing this are not coming at it from this like physiological perspective. And so they're going out and measuring leaf dry matter content, specific leaf area, some of these like seed mass, some of these traits that are really easy to measure and have been shown to correlate really well with these growth strategies. But for me, those don't get at some of these physiological mechanisms. So that's kind of why I chose those specific traits. But again, we kind of just missed the mark. Maybe if we had looked at, we actually do have trait data, I think. So I did actually measure other trait data, right? So I do have seed mass data, and I do have like height data. Those actually just didn't correlate. So those actually would be ones that you would that would be correlated with light competition, but they didn't come out in our analysis as being all that important. So I don't know. That's not a satisfying answer, I know, but I think it just depends on what you're trying to do. Yeah.
So on a similar note, you're working in serpentine soils. So you have a relatively small regional species pool.
Mm-hmm.
So if I were looking at something like if I were working in Great Basin Desert where there's a larger species pool, are there strategies you can think of for how you would reduce the number of species whose traits you had to measure?
I guess so, right? Especially if you were doing this approach where you were trying to find native plants that would be functionally similar to a focal invasive plant. So if you're working in a system, where you have a grass species, like that's your invader, then that probably helps you rule out a bunch of the natives that might not have the same phenology or life form or something like that. But you could also start with some of these really easy to measure traits, like traits that are probably in a database or that you could even get from like a botanical guide and go from there. And then narrow it down and then measure some of these more difficult to measure things like leaf nitrogen content or something like that.
So you didn't have your board of undergrads doing every single one of those traits for every all 53 species?
No, I did most of them. This is a sabbatical project, and you don't have to twist my arm to go sit out in the Serpentine grassland and measure photosynthetic rates. I'll happily do that. But grinding up seeds and foil balling that, yeah, that was all done by undergraduates. I didn't do any of that. Yes?
In terms of a restoration tool, using traits, doesn't this suggest that there isn't one that's similar enough, otherwise it wouldn't have been made in the first place? Or that if there is something that is similar enough, it's not that common on the landscape, so you're actually going to alter the entire community composition by trying to create this other plant and make it more abundant?
Yes and yes. Yeah. So I think that, the fact that these species have come in, like why were they allowed to come in? Yeah, it's a really good point. There is a lot of disturbance in these communities. So I collected the data across, like for the regional species pool data, I actually worked across 2 sites. So at Coyote Ridge, which is one of the sites south of San Jose, that is totally grazed by cattle. And the cows actually do a really excellent job of just zeroing in on all of the exotic grasses, and it actually releases all the natives from competition. I was totally skeptical when I started working in that system that the cows were actually promoting biodiversity, but they do. Jasper Ridge has a bunch of gopher disturbance, and they actually have harvester ants that like completely change the dynamics of that system. So it's quite possible that like the harvester ants are preferentially eating the native grass seeds, which is leading to these imbalances. So I'm not sure, why these exotic grasses have been so successful. But when you're thinking about restoration, this kind of goes back to that philosophical point I was discussing yesterday in my talk. What do you want to restore it to? Like, can we restore it to this reference community from 50 years ago? I think everybody would actually be happier if it was dominated by native plants, but we just, maybe had fewer plants, or we had, more of these particularly aggressive native plants than we used to have in the reference system. So yes, we could potentially be changing the community composition, but I think that's, that discussion should be had, when you're trying to figure out what your restoration objective is. Other questions? Yeah?
So it seems like you're looking for these sort of general correlations between like root ratio and as a trait, as a set of functional traits, but it seems like a more mechanistic approach would be, let's look at light use efficiency, water use efficiency, and model plant growth, and just convert. I mean, you know what the sunlight is, you know where the water, how the water moves through the soil. Why not just model plant growth? And you need to have a much more mechanistic approach for testing how species will grow in wet years, in dry years, with or without nitrogen pulses.
Can you then layer like competitive dynamics on top of that?
Yeah.
Yeah.
Sort of an ecotone model. Yeah, I mean, certainly modeling approaches to model the resource uptake, and then you get a much more mechanistic test.
Mm-hmm. Yeah. See, Noelle, this is why I need to learn how to do modeling. I was just telling her how I need to do modeling. So this is maybe that's the next logical step. So anyway, yeah, I'd be thrilled to look into that. That'd be really cool.
If we have no more questions, please join me in thanking Jennifer Funk for presenting.